Hello, my name is Hannah Willingham, and my study with Dr. White will evaluate prolonged exposure to pre and probiotics in an avian model of treatment-resistant depression. This just means we want to see if our animal model of depression is sensitive to pre and probiotics. This is the first step in determining if probiotics can help people with depression who have not responded to the typical treatments. According to the World Health Organization, as of 2018, major depressive disorder affects over 300 million people worldwide every year. Current antidepressants target the elevation of monoamines in the synapse, norepinephrine and serotonin. The monoamine theory suggests that low levels of norepinephrine and serotonin are linked to depression. However, issues with these antidepressants raise questions. These drugs are known to raise synaptic levels within hours. However, symptom relief often doesn't occur for 4 to 6 weeks, and it doesn't provide relief for all patients. These drugs typically only work for 40 to 60% of patients, and the response rate is about 50%, meaning 1 in 2 people on antidepressants do not have symptom relief. A subset of this population may never respond. This is treatment-resistant depression. The FDA classifies treatment-resistant depression as a failure to respond to 2 of the 4 classes of antidepressants. Considering that suicide is one of these symptoms, this is really unacceptable. The bottom line is that current antidepressants fail to adequately provide symptom relief for all major depressive disorder patients. Another way of saying this is that a good majority of people with major depressive disorder still suffer. The issues under other potential etiological factors and means of relief. One of these areas is the microbiome in the gut-brain axis. The gut microbiome consists of about 100 trillion microorganisms, such as bacteria, viruses, fungi, and protozoa. Major depressive disorder has been linked to low levels of beneficial bacteria and high levels of bacteria involved in inflammation. Inflammation is an immune response to illness or disease. It is your body's way of responding when it's under attack. Major depressive disorder is also linked to a reduction of the diversity of the microbiome. Further, major depressive disorder has a high comorbidity with irritable bowel syndrome, meaning patients with irritable bowel syndrome often also have major depressive disorder. This immune system response has been linked to dysbiosis. Dysbiosis is any change to the gut microbiome and can be caused by things such as stress, diet, and medication. This change in the microbiome can cause depressive symptoms. One way to promote a healthy and diverse microbiome is through the use of pre- and probiotics. Probiotics are beneficial microorganisms, and prebiotics are what feed the probiotics. The aim of the current research is to evaluate the potential antidepressant effects of pre- and probiotics in an avian model of treatment-resistant depression. In order to accomplish this goal, we will be utilizing the chick anxiety depression model. What's unique about the chick anxiety depression model is that it screens for both anxiety and depression while accurately simulating treatment-resistant depression. This model relies on the fact that 1 to 2 week old chicks are social creatures. We house them together, and when they are separated from their peers in the isolation apparatus, they vocalize distress as a means to reestablish contact with their peers. We measure this behavioral data to quantify a DVOC, or distress vocalization rate. The isolation stressor you see on the screen is a plexiglass chamber equipped with video cameras and microphones to detect distress vocalizations. We can test 6 chicks at a time. During a 90-minute isolation period, chicks will initially distress vocalize at a very high rate during the first 5 minutes. The rate decreases by approximately half at the 30-minute mark and remains relatively stable for the remainder of the isolation period. This graph presents mean distress vocalizations as a function of time in 5-minute intervals. The high rate of DVOX during the first 5 minutes represents a panic or anxiety-like phase. The last hour of isolation represents a behavioral despair or depression-like phase. If the chicks increase their DVOX during this phase, we would find this encouraging, as it means the chicks are not giving up. They are not giving in to despair and are continuing to fight. We want them to chirp in this phase. Previous research utilizing this model has identified a unique genetic line that accurately simulates treatment-resistant depression. The black Australorp genetic line shows vulnerability to stress as measured by faster entry into behavioral despair. These chicks were also not sensitive to the selective serotonin reuptake inhibitor fluoxetine and the tricyclic antidepressant imipramine. These drugs are typical of treatment offered to major depressive disorder, and therefore, the black Australorps resisting these treatments is indicative of treatment-resistant depression. To date, this is the only animal model that can accurately simulate treatment-resistant depression. Now that I've described the model, let's take a closer look at the graph. This graph comes from our most recent research done by Roanna Depr and Dr. Stephen White. In this study, chicks were administered pre and probiotics in their food and water for 6 days, with testing in isolation on the 7th day. The blue line represents control animals that receive standard lab diet without pre and probiotics. As you can see, these animals displayed a high rate of vocalization in the first 5 minutes, with a reduction until about 30 minutes and stable during the last hour. This is indicative of behavioral despair. The orange line represents the pre and probiotic treated animals, and while they do show an elevation of DVOX during the behavioral despair phase, statistical analysis revealed that treatment did not reach significance. An antidepressant effect is defined as a significant elevation in DVOX during the last hour of isolation. We want to see a higher orange line. As you can see outlined in the red, DVOX trended towards statistical significance, but did not quite reach the threshold. This led us to believe prolonged exposure will lead to antidepressant effects. Work done in rodent models suggests that prolonged use of pre and probiotics attenuate depressive symptoms, potentially 2 to 3 weeks before any behavioral change can be detected. However, rodent models do have their limitations. First, they have shown a number of false positives, meaning drugs that have passed the rodent models have failed clinical trials. Secondly, rodent models cannot accurately simulate treatment-resistant depression. Based on other literature and these encouraging findings, we hypothesize that a longer exposure may provide antidepressant effects. This timeline illustrates the methods of the study. Male black Australorp chicks will be free-fed either untreated food and water for 14 days, or the treated group will receive food and water containing probiotics for 14 days. As you can see, the previous study done by Roanna Depr and Dr. Stephen White tested on day 7, whereas our study will prolong the exposure and test on days 10 and 14. On days 10 and 14, a cohort from each group of treated and untreated chicks will be exposed to an isolation stressor for 90 minutes. There will be a separate cohort for each group on days 10 and 14. Distress vocalizations will be recorded and analyzed to detect group differences. On days 10 and 14, fecal samples will be collected before and after the isolation stressor. This will help us for later analysis of the microbes in the microbiome. We hypothesize that prolonged exposure to probiotics will result in antidepressant effects, which will be seen in an elevation in distress vocalizations. Additionally, we hypothesize that these behavioral changes will correlate to changes in the microbiome. Because of the link between major depressive disorder and the microbiome, we hope to determine the potential antidepressant effects of pre and probiotics. If successful, this may help to develop novel therapeutics for treatment-resistant depression, and the results could warrant further investigation into specific strains of probiotics. Thank you for your time, and I also want to thank Dr. Stephen White, Roanna Depr, and the rest of our lab. If you have any questions, feel free to contact me or Dr. White. Thank you.